Okay, everybody, welcome to our sixth module, DNA and protein synthesis. Here we're going to learn the structure and function of DNA and RNA. You'll learn the similarities and differences between DNA and RNA, and learn the process of protein synthesis, and create and use models to demonstrate transcription and translation. By the end of this lab, you should be able to review the structure and function of DNA, ID the codons that code for amino acids in DNA and RNA, explain the purpose of start and stop codons in protein synthesis, summarize the steps involved in protein synthesis and define a ribosome and its 3 sites, summarize the steps of transcription including initiation, elongation, and termination, summarize the steps of translation including initiation, elongation, and termination, illustrate and model the processes of transcription and translation, construct a series of tRNA molecules and write the anticodons and amino acids each tRNA carries, and explain the difference in the number of amino acids that were present at the start and at the end of a translation model. So these are your module due dates. Your Module 6 pre-lab survey is due on 6/20/16. Your lab report is due on 6/24, as is your Module 6 lab report summary. You should review the pooled class data on 6/25. You should also have your initial DNA and protein synthesis post on that same day, and your response to the discussion board posts on 6/26. The material that you'll need is shown here. You'll need camera, digital or smartphone to take pictures of your assembled models of transcription and translation. You'll need a pair of scissors. You'll need a color printer, or black and white will do if you don't have color. You'll need 10 sheets of printer paper, a pen or pencil, and Scotch tape. The hands-on lab kit will provide you the DNA nucleotide and RNA nucleotide templates and the tRNA templates. And again, those are files that you can download from the website. Also, you'll need a computer to upload digital camera images, photo editing software such as MS Word, PowerPoint, or other editing software to add labels, leader lines, or text to digital photos. And again, the material from the lecture portion of the course that provides background on what it is we're investigating today. So, DNA is the genetic material of all living organisms, and it's made up of 2 chains of nucleotides wound together in a double helix. The nucleotides are the building blocks of DNA, and they're responsible for the structural units of DNA made up of 3 components in each nucleotide: the phosphate group, the sugar, which is a 5-sided sugar, so we call that a ribose sugar. In the case of DNA, one of the oxygens is missing off one of the carbons, so we call it a deoxyribose. A nitrogenous base, and that's where the information is carried in the nucleotide. There are 4 different DNA nucleotides: adenine, thymine, cytosine, and guanine, abbreviated A, C, G, and T. They're identical in their phosphate and sugar composition, but their nitrogenous bases have different chemical structures, as shown on the next page. The bonds between the sugar and phosphate groups of each nucleotide form the sugar-phosphate backbone of DNA, and the 2 strands wind together as a result of base pairing. Base pairing obeys the ATGC rule of complementarity. An A on one strand will always hydrogen bond with a T on the complementary strand, and a G on the strand we're considering will always hydrogen bond with the C on the complementary strand. The arrangement of the 4 DNA nucleotides creates the genetic code, which is the blueprint of all living things. The genetic code is composed of codons, which are triplets of nucleotides and contain the code for the production of amino acids, which are strung together to create proteins, which are highly complex organic substances that provide a vast number of functions in living things. They include the maintenance of cells, the growth of cells. They are also essential components of living tissue, including skin, bone, and muscle. The 4 different nucleotides provide 64 different codons, 4 options for each of the 3 positions, so that becomes 4 to the 3rd power, which works out to 64, which code for one of the 20 amino acids or a stop codon. And you can see the genetic code here. Okay, so you might say, all right, how exactly does this work? Well, information that's present in the DNA in the form of chains of nucleotides, and so let's make a short stretch of DNA here, right? We'll make up a fictional gene, so AUG, CCC, TAG, and our last codon will be TGA. Okay. And for any gene that we're considering, the beginning of the gene is always called the 5' end, and the end of the gene is always called the 3' end. So the gene is always read in this direction. Now, the information that's in the nucleus of every cell in your body has to be copied and brought into the cell cytoplasm, where ribosomes are performing the task of building proteins. So essentially what happens here in transcription and translation is that we have a cell or the nucleus where the DNA is located. In the nucleus we produce RNA, messenger RNA, by transcription. So DNA in the nucleus used to make mRNA, which is a temporary copy of a gene used for building protein. The messenger RNA goes to the cytoplasm, and the cytoplasm ribosomes plus mRNA will engage in translation to produce a protein. So in our example here of a stretch of DNA, this stretch of DNA will be copied into messenger RNA. So I'll write the mRNA sequence underneath here. The mRNA sequence that corresponds to this strand would be, I'm sorry, this should be a T here, would be AUG, CCC, UA, GU, GA. This would be the 3' end, and over here is the 5' end. And what happens is this will travel out the nuclear pore into the cytoplasm, where ribosomes will attach to this information and begin to use that to build protein, using again the information that's contained in this table. Okay, so in our gene here that we've got, what we do is we read sequences of 3 bases at a time, that's a codon. So this will be the first codon here, second codon here, third codon, and the last codon. Okay? So this protein that would be produced by this stretch of RNA, AUG, would translate to methionine. So it'd be methionine is the first amino acid. Then CCC, well, if we scan real quick and look for CCC, this would be proline. So proline. This is methionine. Then UAG. If we look here, we can find UAG, and that is going to be a stop codon. Okay, so this would be a very short protein, only 2 amino acids long. It would be methionine hooked to proline. Okay, just one example here. Another important thing about DNA is due to the fact that it's double-stranded and anti-parallel, when DNA unwinds to be reproduced, the information in the old strand is used to build the new strand, and as a result, at the end of DNA replication, the same information exists on the 2 new DNA molecules, which each have one old strand and one new strand. And that's the significance of DNA being double helical, is that the information to build the opposite strand is on the complementary strand. As a result, the information is copied relatively accurately into the new DNA that's synthesized right before a cell gets ready to divide. The RNA, as you can see, is a single-stranded molecule made up of, again, nucleotide building blocks, but in the case of RNA, there's no thymine, there's only uracil for those particular nitrogenous bases, and also in RNA we are single-stranded as opposed to double-stranded most of the time. And you can see here the nucleotides that make up DNA and RNA would include these bases hooked to a sugar, hooked to a series of phosphates. Okay, the building blocks are usually triphosphates, and so examples of DNA building blocks would include things like deoxy-TTP, deoxy-ATP, deoxy-GTP, and so on. In RNA, we use uracil instead of thymine, and we're single-stranded. We also don't last nearly as long as DNA does because of our tendency to degrade relatively easily, even without enzyme activity helping. So protein biosynthesis utilizes the messenger RNA to direct what kind of protein we build. It's the process where cells use the genetic code to build the proteins. The process differs slightly between prokaryotes and eukaryotes. In prokaryotes, transcription and translation happen at the same time, whereas on eukaryotes they happen in 2 physically different compartments. One process completes and then the other process begins afterward. In the context of this experiment, we're going to focus on the main steps and the commonalities between the prokaryotic and eukaryotic biosynthetic process, what we call the central dogma of biology. There are 2 main steps in this process: transcription and translation. We start with transcription, which occurs in the nucleus. It's the process by which a single-stranded RNA molecule is synthesized using information contained in a stretch of DNA. RNA, like DNA, is made up of nucleotides with a phosphate, a sugar, which is ribose in this case, and one of 4 nitrogenous bases. Again, the reason we call these nitrogenous bases because they contain nitrogen. All organic nitrogen-containing compounds, with a very few exceptions, are bases, so they tend to lower the pH of a water solution in which they're dissolved. Raise the pH of a water-based solution in which they are dissolved, because they tend to pick up protons. That reduces the proton concentration and thus raises the pH. The nucleotides adenine, cytosine, and guanine exist in both DNA and RNA, but in RNA, the nucleotide uracil replaces thymine and binds with adenine, as shown in this figure. Okay, so you can see here in DNA, A will hydrogen bond with T and G will hydrogen bond with C, and over here in RNA, we see a single-stranded molecule created as a complement to one of the DNA double strands, and it contains the same information, but the building blocks have a few minor differences. So this is a way of getting the information from the nucleus into the cytoplasm. We make a temporary copy of the gene in the form of RNA, and then we bring that out of the nucleus through a nuclear pore, and that's where ribosomes will attach to it and begin the process of translation. There's 3 steps in transcription: initiation, elongation, and termination. General depiction is shown here. In initiation, an enzyme called an RNA polymerase binds to a DNA promoter, which is sort of like the gatekeeper for the gene. This is a DNA sequence that initiates transcription. The RNA polymerase causes the 2 strands of DNA to unwind and separate from each other. In elongation, RNA polymerase travels downstream along the DNA antisense strand, elongating the messenger RNA in the 5' to 3' direction, and doing this again 1 nucleotide at a time. The DNA antisense strand is the template strand from which the mRNA is transcribed. This figure illustrates how transcription creates an mRNA copy of the DNA sense or coding strand, with uracil replacing thymine in the newly constructed messenger RNA. As the RNA polymerase continues to move downstream, the 2 strands of DNA rewind into a double helix. In termination, the RNA polymerase detaches from the DNA and releases the transcribed messenger RNA. In a prokaryote, the released messenger RNA is complete and ready to move into translation, which often happens as soon as the strand is synthesized. Whereas in a eukaryote, the released messenger RNA undergoes a series of modifications called processing before moving into the nucleus through a nuclear pore to be translated by a ribosome. And so, to illustrate some of the differences here, real quick, let's draw a fictional gene and show you how this process will be different in a prokaryote and a eukaryote. So in a prokaryote, the gene is just one continuous sequence of nucleotides, a 5' end and a 3' end, and this would be the complementary strand over here, 5' end, 3' end. What happens is that this will open up and the RNA polymerase will bind to this upper strand and move in this direction and begin to incorporate the nucleotides that are going to be part of the messenger RNA. Once that's complete, this new RNA molecule here will be ready to be immediately translated. So there's the messenger RNA, and then we immediately translated. So ribosomes bind to it, and they begin building proteins out the back, proceeding in this direction down the messenger RNA molecule. So this would be protein, and this would be down here mRNA. Okay, and that's pretty straightforward. But in a eukaryote, it's a little different. In a eukaryote, we have a stretch of DNA. We would unwind it using the top strand here to build our new message. And here we'll use a different color for our message so that things don't get too confusing. Use blue. So our ribosome begins, or our RNA polymerase begins to walk down the strand from this end, and we begin to generate now our messenger RNA, which is going to have some regions that aren't going to appear in the final protein. And if we draw the message, we can represent it, the final messenger RNA, as stretches of RNA, some of which are going to contain protein building information, and some of which will contain sequences that are not going to appear in the final message. Okay, this is all still inside the nucleus. So these little areas here in magenta, those are introns. Okay, so when a messenger RNA, what happens, again, let's indicate here that this is mRNA. Okay, there's the 5' end, and there's the 3' end. But what happens next is that these intervening sequences here, they end up getting spliced out. And so what you end up with is a shorter message that lacks these areas here in pink. In addition to that, we put a cap, a chemical cap, at the 5' end of the message, and we also add a string of adenosines to the end of the message, and so the processed message before it leaves the nucleus ends up looking like this, where the A's, the poly A's, are at the 3' end, the cap is at the 5' end, and now we leave the nucleus and ribosomes will find us. So there's our A tail, there's our cap. Okay, ribosomes will attach. We'll do the ribosomes in black. They'll attach to the 5' end, looking for a place where they're supposed to start translating, and they'll begin to generate now the amino acids that make up the protein by marching in this direction. And for every 3 nucleotide bases, we add a particular amino acid according to the table that we saw earlier. Okay? So that's what we mean by transcription and translation. Translation, which is the second main step of protein synthesis, is the process by which the main messenger RNA, which was created in transcription, is converted into a protein, and a prokaryote translation occurs in the cytoplasm at the same time and in approximately the same place as transcription, while in eukaryotes, translation and transcription occur in different spots. Transcription occurs in the nucleus, translation occurs in the cytoplasm. They're physically separated from each other, and this is carried out again after transcription is completely done, and this happens in the nucleus. There are two major players in translation: transfer RNA and ribosomal RNA, and their overall structure, which is the ribosome, which is the protein building machine in living cells. Transfer RNA is an adapter molecule which is used to bring amino acids to the site of protein synthesis where we can build a new protein. A ribosome is just an organelle which functions as the site of protein synthesis. They're made of ribosomal RNA and protein and are divided into two subunits, a large and a small. The small ribosomal subunit binds the messenger RNA and reads the information contained in this molecule. The large ribosomal subunit contains three binding sites: the peptidyl site, which is called the P site, and the amino acyl site, which is where the nascent chain begins to grow. The new protein is essentially assembled on this part of the ribosome. And finally, the exit, or the E site, which is used to eject transfer RNAs that have already unloaded the amino acid they were carrying. But again, they will eventually be recharged. They'll go back out into the cytoplasm, and enzymes will reload them with their appropriate amino acid. And here we can see again the translation process. Okay, so what's happening here? We're moving in a sequence of three bases from the beginning to the end of the molecule. We're moving in this direction. Okay, a general depiction of translation shows that the first step, which is activation and initiation, occurs when the messenger RNA is threaded between the large and small subunits of the ribosome. The ribosome signals the start of translation when it encounters and binds to a start codon, which always has the sequence AUG, and also has to be in an initiator context in terms of the sequences that surround it, in order to get the protein building machinery to stop sort of moving up and down the molecule with no direction, and find that initiating codon in the right context and begin the process of protein building. So what happens when it gets there is it signals now the large subunit of the ribosome to fully assemble, and then scan the beginning of the beginning of the messenger RNA until it encounters an AUG, at which point the ribosome will stop. The tRNA carrying the anticodon and the methionine that's bound to the transfer RNA will pair up using the ribosome to help it with the codon, and this will be the first protein in the growing protein chain. So it binds to the mRNA codon at the ribosomal A site, creating an initiation complex that signals the beginning of translation. The tRNA bound to the messenger RNA moves into the P site, which brings the next tRNA carrying the complementary anticodon and amino acids to the mRNA, now in the A site. The amino acid in the P site then forms a peptide bond with the amino acid in the A site, releasing the amino acid from the tRNA in the P site and moving the empty tRNA into the E site. Simultaneously, the tRNA in the A site, holding the two peptide bond amino acids, moves into the P site, signaling the next tRNA to bind to the mRNA in the A site. Using this figure as an example, as the empty tRNA exits the E site, a peptide bond is formed between the two amino acids that are held by the ribosome, and as the formation of this peptide bond occurs, the peptide itself begins to grow. Okay? What eventually happens is that we get to a codon that does not code for a tRNA that carries an amino acid, and this is called a terminator. When the ribosome encounters this, what will happen is that it will eventually incorporate what is bound to the tRNA molecule that has the anticodon that reads the stop codon, and in this model that's here. What that results in is that the entire protein is released. It flows out into either the endoplasmic reticulum or the cytoplasm and begins to fold up into the shape in which it's going to perform its final task, which is called its native conformation. At the same time, the ribosome will disassemble. The large and small subunits will detach from each other, and then they may go back to the beginning of the messenger RNA and begin the process all over again if the cell still needs more of that particular protein made. Oftentimes in a single messenger RNA, you'll see an army of ribosomes marching from the 5' to the 3' end, making massive amounts of protein for the cell to utilize as a tool. And this allows the cell to meet its protein quota. Eventually, once we've done that, the protein quota is filled, we'll begin the process of degrading the messenger RNA molecule from the 3' to the 5' end, because we don't need that information to build that particular protein anymore after that point. And you can see how the anticodons in the tRNAs, these are anticodons here, base pair with the codons on the messenger RNA. There's a perfect complementary match. So as we move from here to here, okay, see how the pairing up of the transfer RNA anticodon to the messenger RNA codon allows the correct amino acid tRNA to be brought to the site of protein synthesis and incorporated into the growing chain until we, of course, get to the stop codon. In this case, it's UGA. So in your first exercise, you're going to model the steps of protein synthesis, starting with a single strand of nucleotides and ending with the production of a protein. So you're going to have to print 6 copies of the DNA nucleotide template, 4 copies of the RNA template, and 1 copy of the tRNA template. It's preferable but not necessary to print them in color. The templates are located in the Supplemental Documents folder of your digital courseware. Review the coding strand of DNA in Data Table 1 of your Lab Report Assistant. Create the template strand of DNA and record in Data Table 1. Get your scissors, your tape, and the 6 printed copies of the DNA nucleotide template. Cut the nucleotides from the template. It's not necessary to cut out the entire nucleotide. Rather, cut the nucleotides in a rectangular shape, only cutting out the details of the nitrogenous base ends as shown in this figure. You can see here how these ends that base pair are free, while this is all encumbered by the presence of printer paper. Using the DNA nucleotides, create the entire online double strand of DNA by matching up and taping together the base pairs as shown again in this figure. Take a photo of your completed double-stranded DNA with your name and the data showing in the photograph. Resize and insert the photograph into Data Table 2 of your Lab Report Assistant and refer to the appendix entitled Resizing an Image to guide yourself with resizing an image so that it fits into your lab report. But again, it should basically have this appearance, okay, again depending on which nucleotides are present in your gene and how you've proceeded with transcription. Now, next thing you wanted to do is to determine the mRNA strand that transcription would produce from the DNA template strand and record the mRNA strand in data table 1. Gather the 4 printed copies of the RNA nucleotide template, cut out the nucleotides from the template. It's again not necessary to carry out the entire nucleotide, but rather cut the nucleotide into a rectangular shape, make sure that the nitrogenous bases have enough detail so they can hydrogen bond. Using the mRNA nucleotides, create the mRNA strand by matching up and taping together the base pairs, as shown in the previous figure. Take a photo of your mRNA strand with your name and your date showing the photograph. Resize and insert the photograph into data table 2. Starting with the first mRNA nucleotide, determine what amino acids and codons in the mRNA are coding for and record those in data table 1. Gather the printed copy of the tRNA template and cut out its tRNAs. Okay, so now we're going to take a very close look at what brings the amino acids to the growing polypeptide chain where the ribosome can use them to incorporate into the protein structure. So in our next exercise, protein synthesis, you will model the steps of protein synthesis starting with a single strand of nucleotides and ending with a protein. So build the line of tRNAs that would flow into the A site during translation. Write the anticodons into each tRNA and the amino acid the mRNA codes for. See this figure again to sort of guide you as an example of the tRNA that would be created from the mRNA codons CCU. Okay, so what is the anticodon to CCU? It would be GGA. Okay, so that would be the anticodon loop for this stem loop structure of the tRNA molecule, and it's charged with the amino acid glycine, and so that's what would be incorporated into the growing polypeptide chain at that site. Take a photo of the tRNAs in order with your name and the date showing the photograph. Resize and insert the photo into data table 2. Write the name of each amino acid in the final protein created from your translation and recorded in data table 1. When you're finished uploading your photos and data into your lab report assistant, save your file and zip it so that you can send it to me as a smaller file. Refer to the appendix entitled Saving Correctly and the appendix entitled Zipping Files for guidance with saving the lab report assistant correctly and zipping the file so you can get it to me so that I can evaluate it. Okay, so let's do an example here. Okay, now I don't have so A T G T T T T A CC and TGA. Okay, there's the 5' end, 3' end. Okay, so what we want to do here is transcribe it. So if we transform this stretch into messenger RNA, it would look like this: 5' AUG UUU UAA and GCC TGA here. Here's your 3' end. So, again, what does this make? Well, first amino acid is going to be methionine, again using the info from the table. The next one, which is TTT in DNA and UUU in mRNA, is going to be, look here, let's see, pull up the genetic code real quick. Okay, so here's our protein. Start with the DNA, ATG, TTT, TAA, ACC, TGA. This is the 3' end. This is the 5' end. RNA is going to look like this, where this is the 5' end. AUG, UUU, UAA, ACC, and UGA. That's the 3' end. Now, I have to start building the protein. So this part here is transcription. Next part is translation. So AUG would be methionine. Then we're going to have phenylalanine. And then we're going to have stop codon. Okay, it's easy. Now let's use them to make we've made the protein. What happens to the rest of the messenger RNA? It gets degraded. Okay. And that protein would perform some task in the cell. Now you might say, all right, how do we get all this magic information to build these tools that the cell needs in order to survive? And that comes through evolution and natural selection, where as a result of different combinations of DNA, you happen across a sequence of nucleotides that codes for a protein that provides some advantage to the organism so that it can survive. As a result, their offspring will inherit that same allele, and they have a greater chance to survive than those individuals in the next generation of the population that did not inherit that particular variation. Okay? So, remember again to, when you've finished your translation, okay, you're going to take a photograph of the tRNAs that are incorporated into your growing strand. Okay? The tRNAs are what bring the amino acids to the site of protein synthesis. Take a photo of the tRNAs with your name and date showing the appropriate tRNAs in a photo. Insert it into data table 2. Write the name of each amino acid in the final protein created from translation. Record in data table 1. When you're finished uploading your lab's photos and data into the lab report assistant, save your file and zip it so you can send it to me. And also make sure to include an appendix in your reading entitled Zipping Files if you don't know how to properly do this. Okay? Also, make sure, as always, to watch the other videos that I've included that discuss transcription and translation and how this contributes to cell function. Okay, so that ends podcast 6. I will see everybody in the next podcast where we're going to talk about membrane transport. So, have a good evening, and I'll talk to you later.